In today's presentation, we're going to look at metabolism and enzymes and the role of enzymes in metabolism. So first of all, we need to define this word metabolism. Metabolism. Okay, so it's defined in biological sense as the sum of all chemical reactions that occur in a living organism. And we might think of metabolism in general sense of I have a high metabolism, or I don't have a high metabolism, I have a low metabolism, but in the purely biological sense, sum of all chemical reactions that occur in a living organism. Now these can be further divided into different types of reactions. One of these reactions is what we call anabolic. So it's where we build organic molecules, so going from simple molecules to more complex ones, so building up. And an example of this is in plants and the products of photosynthesis, which is glucose, and this glucose is then turned into more complex sugars and into cellulose material as well, which they used for the structure of their cell walls. And the other type of reaction are catabolic reactions. Catabolic reactions. So these are the opposite, where we go from complex to simple molecules. Okay, and an example of this would be in the process of digestion, where we take in a vast number of different types of organic molecules and we break them down. So proteins from animals are broken down into amino acids. Or starches go through a couple of reactions to break down eventually into glucose. And finally, fats are turned into fatty acids. So in all cases, we take in complex food in our food that we eat, and we turn them into things that are useful in our body. So amino acids, glucose, fatty acids. At these points, they are then go through anabolic reactions, where the amino acids are again made into proteins. So what are enzymes? Well, enzymes are proteins. First and foremost, they are proteins. Now, proteins are made up of very, very long chains of amino acids. Long chains of amino acids, and these long chains of amino acids are what we call polypeptides. Now, a protein might have one or more polypeptide chains. Now, second thing that proteins are, how we define an enzyme as a protein, is that they're globular, meaning they're a three-dimensional structure. And they have a very, very specific shape. Secondly, proteins are catalysts. Okay, and what catalysts are, well, in this case, enzymes are catalysts, and they speed up a chemical reaction. Speed up a chemical reaction. And they also function very, very well at low temperatures. Now this is important because our skin and our tissues don't react well to high temperatures. So we can have a catalyst which speeds up a chemical reaction at lower temperatures and enables metabolism to take place. Thirdly, enzymes remain unchanged at the end of a reaction. remain unchanged. They only need it in small amounts. And so not only do they remain unchanged and needed in small amounts, they can be used time and time again. Now, enzymes are also highly specific, which may basically means that one enzyme catalyzes one type of reaction. So one enzyme one reaction. And finally, enzymes often work with what we call coenzymes. So they may need a coenzyme. So coenzymes are non-protein parts, and these non-protein parts could be vitamins, or they could be metal ions. Now the importance of these is that they bind with the protein part of the enzyme at the active site. And the active site is something that we'll look at in the next couple of slides. And they assist with the reaction occurring. What we're going to look at now is how enzymes work. So I mentioned previously that enzymes are highly specific. So they are specific to one type of chemical reaction. So what we have here is a flow chart of one particular type of model, and this model is the lock and key model. So lock and key model. So we have our enzyme, and we have what we call the substrate. Now these are the reactants. Now, the substrate is what the enzyme acts on. And what we see is that the shapes fit perfectly. And in this first model, it was thought that it was highly rigid. And what we have is an active site on the enzyme that is highly rigid. So the protein is folded in a specific shape to make an active site, which the substrate fits in perfectly. So you've got your enzyme, you've got your substrate, and when they fit together, we have our enzyme substrate complex. The enzyme causes a chemical reaction to proceed, and what we have at the end is the enzyme is left unchanged, and we have our products, C and D, our products. So we might also note in this circumstance that we've got a more complex reactants and less complex products, so simpler products broken down. So in this case, this would be a catabolic reaction. If it was an anabolic reaction, we would have our reactants being simpler, looking just like these guys here, separated, and through this enzyme substrate complex, chemical reaction occurs and the products are actually more complex. So what we're demonstrating here is a catabolic reaction, and anabolic reaction would be the opposite of it. Now if you're looking to model this lock and key method, you could think of a good model would be Lego pieces. Okay, so the pieces must fit perfectly together, the two parts must fit perfectly together for you to be able to build up Lego. The second model came about after testing the original lock and key model. As I said, the lock and key model had a very, very specific active site, and it was rigid, and it perfectly matched the substrate that it was acting on. Now the realization that proteins were in fact not rigid meant that they needed to change this model, and what they came up with was the induced fit model. Induced fit model. What that means is that our enzyme is far less rigid in shape, and we can see that in the crude diagram above, and it doesn't exactly fit the substrate. But when they bind together and form the enzyme substrate complex, the presence of the substrate changes the active site shape. So the presence of substrate changes active site. Now that's very, very important. So the active site has a specific shape but not as rigid. However, when the substrate and the enzyme meet and begin to catalyze the chemical reaction, the enzyme must form a closer shape around the substrate. So it causes a change to the shape of the enzyme to fit the substrate better, and at the end of it we end up with our unchanged enzyme and our products. So we can think of a way you could describe this model would be Plato, the enzyme, which is highly malleable, and Lego for the substrate. So a little bit of a revision of what we've been looking at. We have enzymes, which are proteins, and these are globular proteins, meaning they're three-dimensional and they have a specific active site. Now this specific active site matches one specific type of chemical reaction. Now the enzyme acts on a specific substrate. Okay, so a substrate is the molecule on which an enzyme acts. The enzyme substrate complex is the period in time where, in this case, our induced fit model, the enzyme slightly changes the shape of the active site to better fit the substrate to help catalyze that reaction. And finally, in any reaction, we have at the end our products, and this is a catabolic reaction because we are breaking down more complex molecules, A and B here, into simpler molecules, and we can see that C and D are separate from each other. So as we've already looked at, the role of enzymes in metabolism is to accelerate chemical reactions, to speed them up. Now they do this by not increasing temperature. It's really, really important, because as we looked at, increasing temperature will also damage cells. So the ability to speed up chemical reactions without increasing temperature is vitally important. Now they do this by lowering activation energy. So lowering activation energy. And this process is described in our graph below. So on our graph we can see a pink curve and a blue curve. So this is the progress of a reaction occurring. And you'll notice straight away that the blue curve has a much higher arch on it than the pink curve. As I said, it is all about lowering activation energy. So on our y-axis, it is energy. Okay, anything above this line means that there is energy input needed, and below it is there's energy released. So we can see in this particular graph there is energy being released at the end of the reaction. So a couple of things to point out is both the blue and the pink line, we have our reactants, and at the end of the chemical reaction we have our products. Now the way that these two different chemical reactions progress is all to do with the use of enzymes, and the enzymes catalyze a reaction. So we can see with the blue curve there's much more energy needed, and this is when there is no catalyst. So no catalyst. So what that shows us is there is vastly more, a higher activation energy needed. So higher activation energy for the reaction to proceed. So without a catalyst, the reaction will proceed, but more energy is needed. However, with the pink curve here, we can see that the activation energy is much lower, and that is because of the use of an enzyme catalyst to speed up that reaction. So we have a lower activation energy. The final thing worth noting here is that the use of an enzyme catalyst, does not provide activation energy. It does not provide activation energy, it just lowers the activation energy needed for the reaction to progress. So enzymes have a number of characteristics. Being proteins, they are both sensitive to temperature and to pH. So the first graph we've got here is temperature. So okay, so we can see that we've got a graph, an orange curve, and to label the graph we have a rate of reaction on the y-axis. And on the x-axis we have temperature. We think about the temperature of humans, it's about 37 degrees. So enzymes are going to function best optimally in that temperature. Doesn't mean that they don't function outside that temperature, they just function best at that temperature. So if we think about labeling this graph, we can say that that's somewhere around 30 degrees, and 40 degrees in the middle here, and 50 degrees on the end here. Okay, so what we can see happening is below 30 degrees, enzyme activity is not really basically stopped altogether. At just before 40 degrees, around 37 degrees, we have optimum activity. And past 50 degrees, it falls off very, very quickly after 40 degrees, and past 50 degrees it stops as well. So a couple of things are occurring. First of all, the enzyme works best at an optimum temperature. Outside that temperature, enzyme activity falls away very, very quickly. Once it goes over, say, 50 degrees or more, to be more precise, over 60 degrees, the enzyme actually denatures. So what denaturing is complete changing of the shape of the protein, and it's a permanent change of the shape of the protein. So you think about cooking an egg, the clear, translucent part of the egg turns white. That is denatured, it's changed the shape of the protein, and it can't be reversed. So once something is denatured, it cannot be reversed. However, often when temperature falls below the optimum temperature, it may cease to work, but it can be reversible. It's not necessarily denatured around this lower area. So denatured mainly is attributed to very, very high temperatures. So why is denaturing so important? Or if we remember what my crude drawing of an enzyme looked like with this active site. Now this active site has been brought about because the protein has a specific shape. If we change the shape of the protein, that active site will also change. So if the active site changes, it cannot make its complex with the substrate. That becomes much harder to do. Okay, so if the protein permanently changes shape, the active site changes, and then it can no longer bind successfully with the substrate, catalyzing a chemical reaction. The next graph is looking at pH sensitivity. So pH is a measure of how acidic something is. So if we look at this graph here, we can see we have our pH of a reaction on the bottom. And we can see there's two distinct curves with a little bit of a crossover. Now, and again on our y-axis we have rate of reaction. Okay, so within the cell, most of the reactions occur very optimally around neutral, which is pH 7. Okay, outside of pH 7, most reactions don't work so well within cells. However, in different areas of the body, so the digestive system, especially in the stomach where it's quite acidic, there are enzymes that work and optimally function at very much lower pHs. So we look at this graph here, we start off with 1 through to about 4 and up to about 9 here, and we can fill that in all the way. Okay, very crudely there. So we have two different enzymes, and this is our optimum range for this enzyme. So optimum pH, and we'll call this enzyme pepsin. So pepsin works within the stomach of the digestive system and ultimately needs to function much better at lower pHs. The second curve is for an enzyme trypsin. Now trypsin works on proteins. It works in the duodenum. Since in the duodenum the pH changes dramatically, so it actually works best around neutral or slightly alkaline. So the optimum pH for trypsin is vastly different to the optimum pH for pepsin. So both these work within their own range, and they have an optimum pH. They can work a little bit outside that range, but outside that range they will again start to denature. At the very extremes they will denature. However, both enzymes are active in very different ranges from each other. So in summary, what we see is that enzymes are able to lower the activation energy for a reaction to occur, and to a reaction to occur quite comfortably within the optimum range of the temperature of the body or the organism that they're in. So in the first, the top one, we see that they are active over a very narrow range. Their optimum activity around 37 degrees, and either side of that, the activity falls off very quickly. Extremes of temperature and extremes of pH, for that matter, denature. However, extremes on the cold side of temperature don't necessarily denature, but they will inactivate the protein. In the case of pH, in different environments in the body have different pHs, therefore the enzymes are specific and have an optimum pH within the ranges of those environments. Otherwise the reactions that they catalyze would not be able to occur.